Can you hear me now? All right. Oh, I'm giving you a thumbs up. You can't see me. I don't have the camera on. All right. I apologize. I guess my little L was muted, but Zoom says I'm not. Okay. So all I was saying for those of you tuning in from home, there's variations based off of how some of these protists are able to move. Ciliates with little hair-like projections, pseudopods pushing out their cytoplasm, or the flagellates with one or maybe many flagella, which are longer structures than the cilia. And then I flipped it back here and said there are others, like Eimeria, others that we call some apicomplexans. Malaria fits into that category. And we see similar stuff with sporozoites and merozoites and all of that stuff, just steps in the life cycle. These guys don't have a mechanism for movement. So they don't move, or they move by one of these strategies, all right? That's the idea. So now we're all up to speed. Okay. And every image has this on here. Why? Whatever, I can't even escape out of there. Maybe if I stopped my share, but I'm not doing that. I'm moving on. You'll be left with this smudge, I guess, through the whole thing. You're welcome, right? Technical difficulties. Sometimes you just have to deal with them. I can try to unmute myself, though. That's a technical difficulty that I can fix. Maybe. Okay, so the protists, there are so many different things when we think about protozoal organisms. And we thought a little bit, we talked, Linnaeus with some classification, right? Back before, trying to put a name to a face, trying to deal with all of this. And then we progress, moving forward and thinking about further categorizing things, lending the kingdom Protista into this mix. That's all sorts of different single-celled eukaryotic organisms. But it makes me kind of think back to Linnaeus' time when it was really pretty simple. You're a plant or you're an animal, right? And if you're a single-celled oddball with a nucleus, you're a protist, right? Those protists have huge variation. Some of them plant-like, some of them animal-like, and some of them kind of like a fungus, all right? So really three separate ideas with ways these protists behave, or some cell structures, or whatever it might be, that really makes us think maybe sort of the precursors to multicellular types, like I just said, plants and animals and fungi, right? Where we can see some of these hints in a single cellular form within these. So we are not yet at the point of dividing, but we are super close to thinking these should be separated out a little bit further. And we've kind of got a little of this as we're seeing some of these super groups moving through here, where we've got all sorts. There's other things, animals and fungi down here, but all sorts of protozoal organisms, and they're basically smattered throughout. Right? So pulling them into some separate categories ultimately is right where we are at. We're on the cusp of moving some of these, all right? And we've talked about a few, but we will talk about some more as we progress along. This stuff, man. All right. So as far as the super groups go, that makes me think of like some rock group, right? Excaviata, there are a handful of subgroups within there, some of which we may think of as being parasitic, and a couple, like, you know, maybe not a couple, one right here, Euglena is not, it's a little photosynthetic protist, all right? Often seen in, you know, fresh water, whatever, all right? But a boatload of these cause some very serious infections, right? And some potentially fatal as we get down here to trypanosoma and leishmania, right? So there's some specifics. I'm not going to go through and spout off all of this stuff, but some features that maybe separates them into their subgroups, a few examples, and then potential diseases that they may cause. Giardiasis caused by Giardia, trichomoniasis from Trichomonas, right? And then there's a handful of different types of trypanosomes, some African sleeping sickness, Chagas disease, leishmaniasis with leishmania, and these kind of close cousins, if you will. There's some similarities with some of the infection is really pretty different, right? The chromialveolata, there are a handful within here, and the apicomplexans are definitely some good ones when I think about that, with malaria kind of up at the top of the list as far as pathogens. Cryptosporidium, babesiosis is a type of infection that's transmitted through the bite of a tick, babesia, and Bolinia, that's more livestock stuff. Toxoplasma is another apicomplexan, and it is a pretty common parasitic infection. If you have cats that use a litter box, you probably see warning labels on there. If you don't, you should look at it. Probably some warning labels that talks about caution while you're changing the litter box to avoid the potentials of contracting this, because it is really a pretty common one. And we'll talk more about it later. Some of those dianoflagellates, we can see with these some issues like red tide shellfish that you potentially consume when these big blooms are happening, if you will, and it can lend to some definitely ill effects and some others as well as we move through there. As far as the ciliates go, there are a few that we may think about in like a regular biology lab. Paramecium is a pretty common one to observe. Stentor is another pretty cool one. They have the ability to kind of tether themselves on some sort of substrate, and then they spin around their little cilia. It almost looks like propellers, helping to whip little molecules or bacteria or something into their mouth, if you will, and break it down. They look pretty cool. And I know there's some videos that show some of that stuff that are on the journey to the microcosmos that are, I think, flat out awesome, all right, to see some of them doing their job. So you can see Paramecium on there as well. Balantidium, as far as the ciliates go, it's really the only one we think about for human infection. It causes some GI issues, balantidiasis, definite some intestinal discomfort. And then some plant pathogens, oomycetes. These are some water molds, other things like that, and they cause some issues like that. Right. Rhizaria, there are a handful of different subgroups within there, some amoeboid-like movements within them, and these, a couple fit with some plant pathogens, right? Potato and cabbage pathogens. More algae as we move through here, red algae and some chromophytes. The red algae, Gelidium, is a source of auger, and so is Graceliana, but Gelidium, I think, was the initial one that they started producing this. Kind of interesting stuff. We talked Robert Koch and Koch's postulates and all sorts of stuff before, as we've got this little auger bit right here. Back in the day when microbiologists were trying to grow stuff, they used all sorts of things trying to get bacteria to grow. Oftentimes slices of potatoes, and they try to inoculate stuff on there. Some stuff can grow pretty good, but it also potentially some issues, all right? There were other things using like gelatin, which was a substance that was pretty readily available. You can whip up add some gelatin to some sort of broth and help it to solidify up a bit. This is Idaho Jello. Come on, folks, right? So all sorts of potentials and things maybe could grow on that, all right? But gelatin stuff, when you try to incubate something at our temperatures, when you try to ramp it up to 98.6 degrees Fahrenheit, that's a potential issue, all right? That gelatin doesn't really stay gelled. It starts to liquefy a little bit. You're trying to grow stuff at that temperature, causes problems. And bacteria, if you're trying to get them on a solid media and do that, all of a sudden it's all liquid in your petri dish, right? It caused some issues, some frustrations. And it was ultimately, well, it wasn't Robert Koch. It was one of his researchers, I assume probably a graduate student, really, that brought this to his attention. But really, the story goes, it was that individual's wife. She was the one that knew a bit about agar, right? And agar is a different type of substance that you might also use in cooking, right? It depends on the circumstances. There are some things where you may use some gelatin and help to, solidify it a little bit. Maybe like some whipped cream, right? You could add a little of that in there, and that helps to keep it nice and fluffy longer, right? There are other desserts that you may want a similar sort of thing, but maybe the temperature might be a little warmer as well. Gelatin's not going to do the purpose, but agar does. And this was a substance that some cooks had in the kitchen, probably ones that made some probably pretty kick-ass desserts is what I think, right? They used agar and knew that it would stay solid longer. So this guy's wife knew that. As he's complaining about the gelatin plates that aren't staying gelled, she grabs some agar and says, why don't you try using this? And voila, here we are. It's incorporated into all sorts of types of media, all right? And potentially some desserts, all right? That doesn't mean you should go into the lab and eat the agar, all right? I would not recommend it. It's not dessert, all right? Although some of those plates, I'm like, man, that looks tasty, right? But I bet it's not, right? It just brings me back to Jello, right? I'm like, man, all right. So that's my little spout about agar, all right? Amoebozoa, these are some slime molds, and entamoebas. These are the ones that we think of as some potential issues, and some of them, like Naegleria, huge issues. This is something that pretty much every summer for the past handful of summers, you oftentimes see a couple of instances in the news where somebody dies of brain-eating amoeba, right? Naegleria is the culprit, right? It is becoming definitely more common, and I think part of it's just more recognized, right? With it, and we'll touch on some details with that. It oftentimes doesn't get recognized in that person until they have passed away and they do an autopsy and realize that their brain has kind of been turned to mush, right? And they see these amoebas up within there, all right? Very dangerous. Only a handful, maybe a little more than one handful of individuals that have contracted it have actually survived it, right? You almost always die, right? That is awesome, right? Fascinating, disturbing, and there you go. I feel like right with that, we should stop for the day, right? But we won't. Entamoeba causes amoebiasis or amoebiasis, where it's another intestinal infection. Naegleria infects the brain. Acanthamoeba can infect the brain, but it's a better one for eyes, all right, causing keratitis and potentially lending to blindness. The Epistocontata, there's fungi and animals within this category, and there are a handful of fungal infections that we will touch on, from molds to yeasts, as we progress through. And then lastly, with the animals, we'll want to think helminths, different types of worms, right? So nematodes are types of roundworms, and then the trematodes and cestodes, these are flatworms, right? So tapeworms and flukes, respectively. The trematodes are flukes, and cestodes are the tapeworms. And there's a handful of different things that we will talk about later, right? So this is a slime mold, Dictyostelium discoideum, and there's two images, one where it's on a petri dish and another one, different type, but a similar type of slime mold, Flugio septica, same but different, right? And they move by this sort of amoeboid-like movements, kind of pushing their cytoplasm through, but they also can reproduce as they pull themselves together. They produce structures that we call fruiting bodies, mold-like structures, right? That in turn generate some spores, and there you go. They are not actual mold, right? So here we can see similar stuff in the little fruiting bodies, mushroom-like, releasing these spores, moving back through the environment, going around. We're not, and again, not going to get bogged down with all of the differences in the life cycles, but you can see there are different steps. This is a similar one, where we can see diploid and haploid portions in their life cycle, and different types of replication and morphologies that are observed with those slime molds. So our apicomplexans, remember I mentioned these ones don't have the ability to move, all right? So there are all sorts of different types of these. Here we're looking at just kind of a general, and we showed this sporozoite, an infectious stage of malaria at the start of this chapter. That's what we're seeing right here, right? So we call these protists apicomplexans because up at the tip of this parasite, there's a big reservoir, a big pocket that harbors all sorts of different substances that help this parasite get where it needs to go, to get into specific cells, or potentially to get out of a cell. It just depends on where we're at, what's happening, all right? So as that mosquito, talking malaria, as that mosquito takes a blood meal on us, these sporozoites are within the saliva of the mosquito. And as they get injected into us, as it's feeding on us, there are substances in this apical complex that basically, once it's in our circulation, it allows it to sense liver cells, and our blood is going to be moving through there. It senses that, sweet, I'm in the liver. That's right where I want to be. And it uses some digestive enzymes to link up, not the enzymes, but the cell itself links up with the hepatocytes, and then it releases these substances, allowing it to bore into that liver cell and start this first bit of the parasitic life cycle. And it goes through some asexual replications, and then it gets released, typically exploding that liver cell. And then they target red blood cells and penetrate into them. And now we're in the erythrocytic cycle when we typically really think about ill effects. I think it's very odd that this mark has stayed on every stinking slide, because if I drew on one slide, that should go away, right? It should have changed as soon as I left the slide, yet here it still is. So why? I don't know that. All right. If it's here on Monday, you're like, what the heck? What's going on? All right. So Balantidium coli, these, as far as protozoal parasites go, these suckers are huge. You can see the cilia around them, I think, beautifully. Little fuzzy bits all around it, all right? Our ruler bar right here for 10 microns. Our bacteria are about a micron, right? So this cell is humongous, right? And in turn, as far as those plaguing us, Balantidium is the largest protozoal parasite that plagues humanity, all right? It's kind of a monster, all right? And in turn, as I mentioned in the table of stuff, it's the only ciliate that causes infections in people, all right? So here we can see this ciliated form, trophozoite form, and right there, only one capable of getting us. So paramecium, in this case, these would be something just living out in the environment, not a parasite, right? But there are definitely some details with it. These ciliates, and you know, really with our Balantidium, we might see stuff. We can see probably within here some maybe red blood cells that they have ingested. There's the nucleus, right? I can't quite say where the mouth is, but maybe right here, right? Mouth, if you will. All right, that's this. The mouth, it's this oral groove, all right? And there are cilia that move into that. They move around and help to whip substances like maybe bacteria with our paramecium, or red blood cells, or maybe foodstuffs, because Balantidium coli living in our intestines might just be consuming stuff within the lumen of the intestine. So pulling stuff in within some vesicles, they start to degrade it. There are details in here with the nuclei, macro and micro, with different potentials. We're not going to get bogged down with any of that stuff, but there is some variation as far as what sorts of genetic material is harbored in there. In the case of these ones, there's a contractile vacuole, because think of these living in an aquatic environment. It's going to allow for osmoregulation, and it's not using it like a little jet to propel itself, but it can deal with the water as it's, you know, in this very type of habitat where it can pull water in and express it back out to keep it from being destroyed, right? Because typically we might think the little pond these suckers are in is pretty hypotonic, right? And water wants to kind of continuously be going into these cells, and we want to pump it out, right? So that's the idea with them. Here is the stentor, and like I said, I know there's some videos where you can actually see them in motion. I think these are super awesome, right? So they can potentially tether themselves to some sort of substrate. They can also release and move around. It depends. We can look at these things in a lab. We don't fix them to a slide. We do what's called a wet mount or a hanging drop slide, where a little drop of water with these in there is placed onto a cover slip and a big depression slide. it's got a big divot in there. It's put onto that cover slip and then flipped back around, and the water hangs on the cover slip. And it's like a little world where things are swimming around, right? And you can potentially see some of these and maybe some other organisms that they're potentially pulling in, right? Maybe, you know, feeding on some algae or bacteria. May not really be able to see that, but if it eats a little chunk of algae, you definitely can. A little green bit all of a sudden gets pulled right inside, right? So there's cilia up at the top, like I said, kind of propeller-like, and it whips stuff toward the cytostome or its mouth and pulls it in, all right? So really pretty impressive. And it oftentimes, you know, might just be stationary, and then it senses something's there, and then the propellers start going. That's pretty cool. Here's an oomycete, a water mold, a saprobe, some saprobic organisms, or a saprophyte, are organisms that grow on or feed on dead and decaying material. So we think of these as, you know, potentially playing super important roles in nutrient cycling. Saprobic organisms, molds are saprobic, right? And this basically, you can't see it, but apparently there's an insect underneath there, and it's covered in this water mold. Here's our euglena, and like I said, some, you know, photosynthetic capacities with these chloroplasts within there. They do, you know, in their aquatic environment, they do want to kind of recognize where they're at. And so they've got this little eye spot that helps to kind of sense in that water table where they truly are. And in the daytime, they would like to be more toward the surface, so they could be photosynthesizing. And that eye spot helps them to recognize truly if they are moving toward the surface or going toward the bottom. So these are a flagellate. You can't see the flagella up on this, but flagellated. So the trypanosomes we've mentioned briefly in that table, couple of different potentials, sleeping sickness or Chagas disease, couple different species. Really, there's a handful. Trypanosoma brucei is the culprit for sleeping sickness. Really, we've subdivided it. It's a subspecies. There are three, and if we looked at them under the microscope, you couldn't tell. Right? They all look the same. But where they reside and whom they potentially infect can definitely be different. All right. Trypanosoma brucei infects livestock, causes sleeping sickness-like symptoms in those animals. We call it nagana. The other ones, the other two, are what plagues humans. And those Trypanosoma brucei gambiensiae, and the other one is rhodesiensiae. The Gambian and Rhodesian forms, right, respectively. And there are some varied types of infections that we see with that as well. One of them, and we're not going to get bogged down with all of this right now, one of them is more important when we think about actual central nervous system involvement. It's one that's a little more chronic, if you will, and the other more acute, and you ultimately die before you ever get any sort of brain involvement, all right? Not going to worry about that right now, all right? So the African trypanosomiasis, or sleeping sickness, there is a fly that transmits it called the tsetse fly, taking a blood meal, inoculating the parasite into us through that blood meal, right? They in turn start replicating by binary fission. These live with the cells, not in the cells, red blood cells, right? And we can see them also in lymphatic system and spinal fluid, if we're talking the one that causes really the sleeping sickness, right? The tsetse fly takes a meal, ingesting a different stage of these trypanosomes. There's a couple of different replications and divisions that happen inside of us, getting it ready, letting it develop, if you will, to be ready for that tsetse fly to feed on. And there's a couple of rounds of divisions, maturation within the fly to get it ready to be infected into us, all right? So it's multiple steps in both hosts for that to be occurring, all right? All right, and I truly think really pretty awesome looking parasites, disturbing, but still pretty cool. All right, shifting over a bit away from the protists to some bigger stuff, potentially bigger stuff, all right? Moving to some multicellular, the helminths. There are a lot of potentials with these. Enterobius vermicularis is called the pinworm. They also sometimes call it the seat worm. It is a parasite that is really pretty common, right? It's one that, you know, you probably don't want them, but I like to think if, you want to get some worms just to say you had them, pinworms are probably the better ones to maybe get, because they're really, although you might get a little bit of itching, you might get a little bit of irritation. There could be some other low-grade problems. It's not something that you're super concerned about, not something that we think of as a deadly or fatal type of infection. Disturbing for sure, all right? It's a pretty good one for little kids. Daycares and situations like that may lend to a greater opportunity for it to be spread amongst little kids. Same with schools. They become kind of a breeding ground for all sorts of things, and the eggs can be contaminated kind of all over the place. And that's what is infective ingestion of that egg. It hatches out in the intestine. The nematodes are dioecious, separate sexes, right? When we get to the flatworms, they're hermaphroditic, right? Most all of them, all right? So the separate sexes, they find one another, they pair up, they mate. Almost always, the females are quite a bit bigger than the males because they're going to be producing copious amounts of eggs, typically. And the male, he's got one job, right? Got to fertilize. And basically with these, as soon as that happens, once copulation occurs, he is spent and dies. Done deal, all right? She's going to help, these eggs get out of the body, and shortly after oviposition, depositing these eggs around the anus, she doesn't have much time left either. Pretty quick life cycle with them, all right? Depositing those eggs, and there's potential maybe for a couple migrations to deposit eggs, but typically once that happens, they're dead as well, all right? So this, in turn, is a pretty easy parasite to be transmitted through contamination situations or reinfection. So it just kind of keeps it going within you if you have it, all right? They are really very little, lending to the pinworm name, all right? Oftentimes I think about worms kind of being huge, all right? But pinworms are not, all right? Couple millimeters maybe, all right? And we'll talk some details of them later, all right? There's so much that I'm like, I want to spout off more stuff, but I know I'm going to talk about them again, right? You don't want to hear it all twice, right? Maybe. It's good. Okay. There are moving from those roundworms to the flatworms. There's a handful of different types, all right? And we can categorize them into some classes, the turbularians. These types of worms are free-living worms, oftentimes marine, but it could be some freshwater environments as well, where we may see some of them, all right? Monogenia, these worms, the monogenia means a single host, and oftentimes it's insects, it's arthropods where we typically see these guys infecting, or maybe fish, right, gill flukes. The next ones, both trematodes and cestodes, we call these digynians, two hosts, and in some instances it might be more than two, all right, but two, all right. Trematodes are the flukes. These are kind of leaf-shaped or oval-shaped, and there's a handful of different ones. Fasciola hepatica is a liver fluke. And this next one, the tapeworms, cestodes, these are long and segmented. The segments we call proglottids. Both are hermaphroditic, all right? So a single worm harboring both egg and sperm, and for the most part, it only takes one for them to produce viable eggs. There are a few kind of rarities maybe that require cross-fertilization to actually produce viable offspring. Even though they're hermaphroditic, they need to be fertilized by someone else, right? But most of them, it only takes one worm. In all circumstances with these, we don't think of them as replicating inside of us, and you get infected with one tapeworm and then all of a sudden you've got 1 million tapeworms in you, all right? You get infected with this one worm, it's replicating and producing eggs that you defecate out, but they're not ramping up their populations in us, all right? With the pinworms, it's the same idea. They're not ramping up their populations, but you do have a bigger opportunity potentially for these eggs to be ingested and keeping it going. It also, with the high contamination issues, I like to think of pinworms as fun for the whole family, because if you got a little kid that's got them, you probably all have them, right? And you don't want to think that. You're like, I'm going to keep my kids outside, right? That's where they should be, right? All right. So here, thinking the tapeworms, in this case, we are looking at Taenia solium. This one, saginata, beef tapeworm, solium, pork tapeworm. And they look pretty similar, but the pork tapeworm's got a bunch of hooks up here at the head region or the scolex, right? And saginata doesn't have that. It's just got some suckers. And these suckers are just holdfasts, attachment devices to get them adhered to intestines. They're absorbing nutrients right through their skin, their tegument. Opisthoorchis viverni, this is a fluke or a liver fluke. And it also has some suckers that allow for attachment. But again, they're going to be absorbing through their skin. This is a good image because you can see some of the structures within it, and almost all of the inside of these parasites are reproductive structures. Massive amount of capacity. A lot of this dark stuff are reproductive structures that we call vitellaria that are going to be producing like the yolk-like material to nourish these eggs. Down here, likely the testes. There can be numerous testes, typically a single ovary, and maybe a dozen testes, right? Kind of crazy. All right, I think we will stop with that. We will pick up on Monday with schistosomes. Farewell, good day, have a great weekend. I'm sorry I started this with no volume.